And welcome. Hello. Hello. Hang on. Let's see. Can you say something for me, please? Yes. Oh, fuck. It's always fucking audio problems. Okay, I think you can hear me. Everyone should be able to hear me, but I don't think probably is. Don't think so. Yeah, I think it's you. Okay, say something.
Hello. How's everyone?
There we are.
Much better.
Okay, can you say something again?
Absolutely. What would you like to hear?
Your beautiful voice, especially who you are, where you are, how are you, and why are you?
I'm Dr. Stephen Hobbs. I'm here from West Texas, enjoying life out here in the sun, at least trying to. The sun's starting to go down now. It's about 6 o'clock. So, yeah, going down. But we got beautiful, clear skies, so plenty of stars tonight. It's going to be great.
Oh, nice. So, because you are an astronomer, chemistry, physics, micro, you're literally everything, aren't you?
I try to be a living science book.
Yes, we need more of them, actually, don't we?
It's great. I love it because I get to talk to everybody.
Yeah,
like if you're a scientist on the planet, I can probably talk to you.
Well, I'm glad to have met you. So, I also have to say a quick thank you to Mama Atheist because she was like, Mero, get him on, and I'm like, okay, I'll do that.
Crazy guy? Okay, sure. I appreciate that. Thank you.
So I also want to say a very big thank you to, of course, Zolfner for the $5 super chat already. You are brilliant. No. That's good. Also, a quick hello to Purple Rhymes of Orange, Beach Price, me, Mama Atheist, Critical Cripple, Atheist Edge, hello, and Almost a Doctor. Almost a Doctor is also another Australian walking science book. So if you wanted to have a chat to Almost a Doctor.
Absolutely.
Yeah, so anyway, I will be able to increase. How can you say something again?
Absolutely.
How's that?
Yeah, we
Also,
are very excited about this. I say we. I know I am. I can see from your face that you are as well. So
I am.
I get to talk about one of my favorite subjects.
Okay. So if anybody has any complaints about audio, just yell at me. Okay. It's okay. It's fine. It's fine. I know I'm crap at streaming. All right. So the reason why I'm super excited is because I have a passion for microbes. And of course, Dr. Stephen has also got a passion for microbes. And when two passionate microbiologists sit down in the same room for,
Any
10
length of
minutes,
time.
any length of time, we have some pretty fascinating conversations.
Absolutely.
So, my favorite microbe is Gluconacetabacter kombucha, which is a very cool little microbe that produces cellulose. And I won't go into that too much because I want to hear more about Dr. Stephen's favorite microbe, which is
I want to hear more about that. It's
okay.
Mycobacterium tuberculosis.
Mycobacterium tuberculosis. So,
Yes.
if you don't know anything about the naming of bacteria, a lot of the times our common name actually is rooted in the scientific name. So Mycobacterium tuberculosis causes the disease tuberculosis. And Gluconacetabacter kombucha is also the bacteria that actually produces the fermented tea kombucha. You know that the meme of that woman taking the sip of the drink and she's like, oh, and then she's like,
Yeah.
That's kombucha.
It's not bad. It's not good. It's pretty good. Not bad.
Yeah, no, I quite like it. It's definitely an acquired taste, and or it's either you love it or you hate it. I mean, it is pretty gross looking. I will not lie to you about that.
Yeah, it's got stuff in it. I don't know if I can drink that. I've tried. No.
Yeah, that's fair enough. That's fair
It
enough.
reminds me of my lab. Yeah, I'm supposed to drink that.
Reminds me of my lab too. So, yeah, so I do want to have a quick chat about kombucha because there's some really cool things going on about it. For example, as I said, it produces cellulose, and what cellulose is like a sugar. It's a structural sugar, and it causes what we call a pellicle right on top, forming on top of the tea. So you boil tea, make it really sweet, pour it into a jar, and it will just sit there, turning the kombucha, the sugar, the really sweet sugar, into acetic acid, which we know is vinegar, and carbon dioxide and alcohol. So it's very, very slightly alcoholic. It's quite fizzy as well. And if you do a second ferment, you can actually have it go explodey.
Right.
Yeah. So yeah, you got to be careful. It can be a little bit dangerous like that. I've actually had a Grolsch bottle. One of these. These things are rock solid. They're really, really thick. I've had it explode one of these. So they're pretty big. They're pretty explodey. And just sprayed myself with kombucha.
Is that free once you have one micro lab?
Anyway, but one of the really cool things about this is it can produce that cellulose, and there's a lot of different things that that they're looking at making. One of them is being a scaffolding for nerves.
Okay.
So peripheral nerves, and they're looking at using that to maybe potentially use as scaffolding to make what's it called, to help
Axon
fix help fix your peripheral nerves. If you've got peripheral nerve damage, they might be able to use that.
I actually posted that today.
But what was that? Sorry,
I actually posted that today. Some of the nerves that I've been growing here in the lab, I'm trying to direct their growth. So knowing that, I can add to it.
Yeah, so that's pretty cool. There's also bioremediation that they've been able to do. They've been able to use carbon kombucha as a way to encourage the remediation of rare earth metals, which is a major environmental issue.
Absolutely.
There's also a potential that I particularly want to go into is investigating in vivo use of to stop H. pylori, which is Helicobacter pylori, which is probably one of the most famous bacteria that we have because it produces H. pylori causes gastric ulcers, so stomach ulcers, and also it can lead to all sorts of nasty things like cancer and things like that. And so one of the advantages to using another microbe to fight another microbe is that we don't have to rely on antibiotics because there is an increase of antibiotics resistance. So kombucha is a pretty cool little microbe. I really, really like it. And same with H. pylori, but I've talked too long about bacteria. Can you tell me something about tuberculosis?
That's okay. No worries. Mine is not nearly as fun as yours.
Yours just caused death and
for
disease.
A pretty fun drink, and mine is responsible for 250 million deaths. So yeah, I mean, I focus on the pathogen part more so than all the cool bioremediation, which I love. We have super funds here in the United States, and we just dump as much money into it as possible, doing research to see if we can prevent some of those, environmental damages. And a lot of them actually have bacterial projects going on right now. This being one of them with rare earth. So I love it. Mine, on the other hand, is feared. Like, we don't want tuberculosis. No one does. Yours, we'll take it and put it in a sprayer and spray it all over and hope for the best. So yeah, this one, we're trying to prevent it completely. I would love to have some type of antagonism. I would love that. But we don't have, a lot of our immune systems can't keep track of it. And that's where we get the secondary or rebound effect with tuberculosis. So, and that's where a vaccine would come in perfectly if we could make one.
If we can make one. Yeah, making a vaccine for a microbe is vastly more difficult than making a vaccine for a virus. Yeah.
Go ahead.
Oh, no, you go.
Oh, no, go ahead.
Okay. I was going to can you explain why making a vaccine for a bacteria is significantly more difficult?
Yeah, going around the outside of most bacteria is a polysaccharide, or a related molecule for that. They can change quite often. Surface receptors, the actual chemical makeup. A lot of times they'll outsmart us. Like, if you don't get all of them, that one that has the small thing that's different will actually start replicating. And that's where we get antibiotic resistance. If we've got something like a vaccine, we'll make it against one, but there's already another strain that exists. We just don't know about it yet. So we're thinking that we're fighting one. We've actually got 2 or 3 or 4 or 5. And then it becomes resistant, or they become resistant. Yeah.
Titan, who I also like to call Giant Butthole, says isn't one of the problems for vaccine for TB is that there's so many variants. So
yes.
Also, very quick hello to all the people who've just popped in. Giant Butthole, Lassus Vulgaris, I'm sorry. Don. There we go. Brandon Worrell. I think I did see Dr. Osiris. J. Mike. If I have missed you, yell at me in the chat because I apologize. Oh, no dancing palm trees. There we go. Also says that they are not a big fan of contracting tuberculosis. I would agree with you on that one.
Yes, I am right there with you. So we have a history here. The reason why I love it and I hate it at the same time, I love it because it has bounced around so much in history. I hate it because it's killed so many people, people that I know, patients, everybody. Almost every one of us are affected in some way, fashion, or form. Like my granddad, he actually died of tuberculosis in 1943,
Wow.
right after my grandmother, or my great granddad, right after my grandmother was born. So there's a lineage there. She doesn't know anything about that. We don't have any paperwork where he was from. So that lineage, other than going to him, stops. I have no idea. And then the HIV epidemic, AIDS epidemic in the 80s, watching people that would, have HIV contract multi-drug resistant tuberculosis, it was horrifying. So we were losing 2 million people a year, from 1984 to 1992. So I grew up watching this stuff just move through the population rampantly. Yeah. So I started studying it. It's like, why is this showing up? Why is it still here? How is it still here? And so on. And I just kind of fell in love with studying it. And then I wanted to, eliminate it completely, which I know will never happen because it has other variants that get into other animals. We get from cows. So it's relatively common in the animal kingdom, but I want to get it out of humans as fast as possible, or at least find a successful prevention.
Yeah. Linda Bell actually asks a really good question. Dr. Stephen, my mum was a nurse in the World War. During her training, she had to test positive for tuberculosis, but dormant to work on the TB wards. What does that mean?
All right. So that means most likely she was exposed, and only about, I would say, somewhere between 35 and 50% of cases become active a second time. So you do get exposed to it, but it gets locked up in a granuloma inside the lungs. So what a granuloma is so you've got these little bacteria that are in there. The macrophages try to eat them. They call in for reinforcements, T cells and things like that, and they wall it off with these fibers. Most of the tuberculosis cases, it stays in that granuloma. It doesn't come out. In some patients, and that's where it becomes, miliary TB or something like that, it becomes an active case. It becomes a necrotic granuloma, and it breaks open. That's where it can spread to other people. It starts doing damage. It's already evaded your immune system at that point. So as long as you're not there, you don't have an active case. It's latent, and we're hoping to keep it that way.
Yeah. The interesting thing about Mycobacterium is that I can't remember it doesn't have a fixed shape. Like it's not a coccyx, which is like a circle or rod shaped. Did you want to talk a little bit about that? Because I'm trying to dig through. Like last time I was in a microbiology lecture was like 2 years ago. So I'm just going, what was that word again?
Oh, sure, yeah. So the peptidoglycans that we typically find on bacteria make them have a particular shape, as you mentioned, cocci and bacillus and spirals and so on. This doesn't have that. It's got a very waxy coating on the outside. So for a really long time, even after we learned how to pick out other strains of bacteria, we could not do that with tuberculosis. Robert Koch is one of my heroes. I love it. I could read about him all day, every day. If you don't learn about any other scientist, please learn about Robert Koch. So he comes along, he finds a way of identifying it. We can't see it very well. Okay, so we have to find some other way of staining. He sees it, he can see the effects, he knows that it's there, but he doesn't see it very well. So it took a little bit for us to rediscover how to diagnose tuberculosis on the cellular level. My brain has just jumped the track. Sorry, I'll get back to it. Where was I going with that? I am so sorry, guys. I've lived with this.
Robert Koch? Figured out Gram scan, not the Gram scan.
That's where it was. The cellular structure, that's where I was going.
Yeah.
So the cellular structure has something that's called mycolic acids, and that's really where we get the name Mycobacterium. Mycolic acids are found there. Those mycolic acids have to be stained a completely different way, and it's called acid fast. So we have, Gram stain for some bacteria. We have these other bacteria, including tuberculosis and leprosy, that we have to acid fast stain them. So they're either acid fast or not. And they stain this really bright red. It's really kind of cool. I'll post a picture of it on Twitter if you'd like me to do so. I've got some right there.
Would you be able to spell his name?
Koch? Yeah. I'll send it to you.
Yeah. Do you want to do it in the
Yeah.
chat?
Oh, well, it's there, but it's Koch.
Oh, you're Dr. Osiris. Oh. No, you're not Dr. Osiris. No. Coach. Okay, I'm confused. You continue.
So it's Koch. He started off as kind of a country doctor and wanted to know everything possible. I mean, everything.
Mm-hmm.
And he was. I sing his praises, really. I mean, he was brilliant. Everything that he did with a purpose. And he did it not so that he could take over or anything like that. He did it because he wanted to know everything about everything. But they didn't know everything about everything, so he invented it. That's what really just powers me studying him. How did he become so inventive so that he could have his fingers in almost every pie possible? He was brilliant. So they end up, having the Koch Institute and so on. And really and truly, any of the treatments that we have for bacteria, I'm going to go ahead and just, it's attributed to him. It's his work.
Yeah.
Brilliant.
Yeah, it's a bit like that with Lewis and chemistry. So, he figured out the Lewis dot structure. He figured out the Lewis acid base definitions. He was big. He was nominated many times for a Nobel Prize, but never got it because one of the people on the Nobel Prize didn't like him. Nobel. So because of that waxy surface of Mycobacterium, it's almost impossible to kill, basically.
Yes.
Yes.
Another cool thing about them, I say cool, it's terrible. So understand that I'm going to use those interchangeably. Something cool about tuberculosis is that they are really patient. Really patient. So they can outlast you in your patience. So you'll take, a couple of antibiotics here or there that were developed in the 1950s. They would just sit there and bathe in it, just waiting. It would go away, and then they would replicate again. So you would think you were treating it the whole time, and then the bacteria are just sitting there laughing at you, or whatever patient. After you finish taking the 1 week, 2 week, 3 weeks round of antibiotics, it was still there.
Yeah.
I love it. I mean, that is an evolutionary nightmare and brilliant at the same time.
Yeah, because
we can't treat it. We can't now, but
because it's a spore forming bacteria. So a spore forming is basically when a bacteria's and also if I get the name wrong, as I said, last time I was in a microbiology lecture was a couple of years ago. But spore forming means that basically it has a pocket of its DNA that sort of like gets stored away inside the microbe. And then if it gets hit by something like desiccation, or really hot temperatures, or if the bacteria itself gets stressed, it will die, but the DNA, the little pocket of DNA, is protected by by the spore, and so once the things become more favorable, the bacteria just reinvigorates itself. Because that was something I remembered about TB as well. So by the mycobacterium, genus.
Yeah.
Beach Price says, how long has TB been in human populations?
Since before the last ice age.
Jeez.
So I say right at the very beginning of the last ice age, as animals and humans were, getting together a little bit more, as humans were migrating, we actually find damage from Pott's disease, which is like bone tuberculosis. We find that damage in a lot of the artifacts from the ice age and just before.
Wow. It's probably been around for a bit longer than that. It's probably been around basically forever. It's a very old microbe, isn't it?
Very. I read not too long ago that we find it in some of the larger of the bison species, which means it's been around in cows and bison forever. So That would be a good research project to find out exactly when, but you've got to find those artifacts at the same time.
Yeah. There's a type of disease. It's the only disease that can get jumped from domesticated fish into humans, which is Mycobacterium pescina or something like that, which is known as fish TB. So if fish have got TB, there is a good chance it's been around since ever. So yeah. And how many people still die today from TB? Says Jen with one N. And hello, by the way.
In the United States, it's rare. Everywhere I say it's rare. It's happening more and more. It is becoming more of a problem because of the multi-drug resistant. And I know that not everybody lives in the United States. So worldwide, about 12 million people a year.
Jesus Christ.
It's estimated 12 million people a year die from TB.
Yeah.
So any advance, anything at all that we can get over and above what the bacteria can do, even if it's just preventing it from spreading, saves lives.
Yeah.
So
wow.
Test. Okay.
We don't have it in Australia. There was something that I learnt at uni which got me kind of cranky was there is a hospital, I think it's on Christmas Island. It's on one of the islands off the Australian mainland that specialises in protection from, specialises in treating tuberculosis. Mm-hmm. And the Gillard government, which generally she was actually quite progressive, but they actually cut funding towards that hospital. And I'm like, are you fucking stupid? Like, jeez. I was like, why would you do that?
Yeah. We have a few of those treatment facilities here in the United States. Those in the United States are not required to get treatment. It's one of the questions about legality in medicine. Does one person's personal right trump the entire public health? Like if you've got COVID, you can go to Walmart. Would they be able to arrest you and quarantine you? All of that came out of and it's the Typhoid Mary study. I hope everybody knows who and what Typhoid Mary's story was.
Yeah,
but New York originally lost that battle. We put her on. Yeah, we put her on the island and said, you can't do that. You're willingly spreading it. And she sued New York and won. They said, okay, we will release you, but you cannot work as a cook. Like you're spreading it around, killing people. And she said, okay, I'm not going to do that. So she changed her name to Mary Brown and worked for a hospital, killed some more people. And then when they caught her the second time, she's like, yeah, I'm just going to live on the island.
Yeah. I feel sorry for her, actually. She's very much hated, but at the time, germ theory wasn't hugely accepted. She was uneducated. There was no way for her to make a living outside of being a cook. She's been demonized a lot by history. Everything.
So yeah, her name is Mary Mallon. If they want to look her up, it really is a good story. Even more so knowing that she punched 4 cops and won.
Nice.
Like they went to get her and she kicked their asses.
She's one of my heroes now. She's so good. So yeah, so Mary Mallard, please be more respectful towards her.
Of course.
Yes.
So with TB, if you have an active case of TB in the United States, there can be steps taken so that we can quarantine you in places like state hospitals. We don't normally do so as long as somebody is taking the medicine like they are supposed to, but unfortunately they don't, because it takes about 18 months really to successfully, completely treat tuberculosis. What's the likelihood they miss a dose in 18 months?
About 100%.
Yeah, they're going to. So we need to have directly observed treatment, DOT, or doctor observed treatment, which means we have to watch them swallow the pills.
Yeah.
You can't do that if they're just living down the street. So a lot of times they'll go to a treatment facility for about 18 months and stay there. Wow. But the good thing, we do have treatment facilities.
That's true. It just would be extremely expensive, that's all.
Yes. Yes, it is.
Yes,
Um,
because
Another side story to go with tuberculosis. I lived in Dallas for 8 years, and do you know who? I don't know, but the stories that pass from one country to the next. I recognize my audience, but do you know who Wyatt Earp was?
who?
Wyatt Earp, Doc Holliday. Do
No.
either of those sound? It's okay, no worries. Those in America are screaming right now. Yes, I know. So Doc Holliday was a dentist. That was his original career plan, and he got tuberculosis from it. So he becomes, this kind of bigger or larger than life character because of Wyatt Earp and trying to take over the Wild West and so on. They wanted to bring the rule of law into the American Southwest. Those of you who are historians, I understand. Hang in there. Well, Doc Holliday's office, where he contracted tuberculosis, was in Dallas. And at the time, they were putting up a placard when I got at the apartment that I did downtown, and it was the exact same one. So I was freaking out at the same time because I was moving into what used to be Doc Holliday's office. That's where he contracted the thing I'm studying.
Oh wow, that's so cool. That's really cool.
Anyway. So sidetrack.
Sidetrack. Yeah. Well, yeah, because we did briefly touch, but I'd like to go deeper into vaccinations in regards to microbes. Because, we did touch on it because you're also studying the COVID-19. You're also doing some research into that. So I'd like to talk a little bit about the difference between making a vaccine for a microbe versus a virus.
Yes.
Because one of the big things that people are saying is that the COVID-19 vaccine came out too quickly. It was too quick, but I'll let you talk about that because you probably understand it better than I do. So,
really and truly, we have been studying coronaviruses since the 1960s, and we've been working on a vaccine for them for about 30 years.
Yeah, it's not particularly new technology, is it? No.
Yeah, no. But with that statement, we have come up with a lot of new technology since then. So a lot of times when you read, a science book, the last time that was cited was in 1962, but it's in a brand new science book. The technology has continued to change and update and just grow exponentially since then, really. So coronaviruses, I've had them here in the lab, coronaviruses multiple, so that I could study them. Okay. Well, I've been doing that since 2012, and I know that if I've been studying them since 2012, people have been sourcing them since long before then, and there is a reason we have been studying that. Outside of this particular strain, usually what would happen with coronavirus is you would get a cold. That's originally where some of this misinformation was coming from. Oh, it's just a cold. It's not. COVID is not just a cold. They are related to each other. It's kind of like Jeffrey Dahmer's uncle is not Jeffrey Dahmer, who's a mass murderer here. Do you know that? Okay. All right. So not the same, even though related. Okay. Well, since we've been studying these forever, what we've been trying to do is find one for all of them. We've been trying to find one vaccine for every coronavirus because that would stop not only COVID now, but the coronaviruses completely. All common colds are saying 50%. Sorry, about 50% or so of the colds that we would get would just go away. So downtime, productivity, people not getting sick, the related respiratory cancers and stuff, all of those effects would drop off with just one vaccine. So we've been working to find one particular part, the spike, or a strain of mRNA that would go along with that. It's taken us a little while to do so, but when I say a little while, I had originally planned, or I projected when COVID came out last year, that we would be probably April or May before the vaccine was available. And that was pushing it. That was about 18 months or so, really, since the first reports of, at the time, was Wuhan virus. And we knew how bad it was. After the World Health Organization named it, they came up with the name for it, of course. COVID. They said exactly what it was. We had already been able to pull out their DNA, or the RNA, and read through it. Everything was already ready. We already had technology to read it to make the vaccine. So they started almost immediately ramping up, and you have to go through all of your paperwork to make sure that you can do, human trials. Some of those had already started, but not on COVID, on other coronaviruses. They just switched the code, if that makes sense.
Yeah. Also, if you haven't seen my video on why calling COVID-19 anything other than COVID-19, please go and watch it. It's actually one of my favorite videos I put out, and also one of my more important ones.
Absolutely.
Yeah. So, because it is a little bit racist, despite what everyone keeps saying.
Absolutely.
Yeah. So just be aware.
I'm there with you. Same thing. I'm the same with the Spanish flu. I don't like it at all. I call it the Great Flu of 1918. There's no other reason to call it something else.
Yeah, especially since it didn't start in Spain.
Yeah, it started in Kansas.
Yeah.
Yeah. So, but yeah, I'm right there with you. The only reason I mentioned it is because that's what it was at the time before the World Health Organization was able to name it. But that was the first thing, really and truly, other than stopping it. Like, how do we stop this? That was the first thing they did. Name it.
Yeah.
Yeah. So highly racist. It bothered the crap out of me for a long time that Trump was still calling it, something else.
Yeah. And that's because it was racist.
Yeah, on purpose. He did it on purpose.
He did it on purpose. It bothers me. That's why I ended up having to make that that video. I got all of that information from a podcast that I love listening to called Skeptics with a K. It's from the Merseyside Skeptics Society. There's a lot of different skeptical thinking, science-based, science communication-based podcasts that I really like listening to. It's that one, Skeptics with a K, and also Skeptic's Guide to the Universe. If you want to listen to some really good skeptical podcasts, listen to them.
Awesome. Thank you.
Yeah. Almost a Doctor does ask is, how do we know that the 1918 influenza pandemic started in Kansas?
We can follow the records back. We see exactly what was killing them then. One of the things that happens with flu, or pretty much any virus, is where does the outbreak start and where were people keeping track of it? The most important aspect that occurred in 1918 was that people were keeping meticulous records on military bases. They were calling for more resources, saying something is sick, like something's causing us to get sick in very large quantities. They're saying you can't do that. So put them all together. But if you put them all together, you're going to spread it, and it spread even further. At the same time, we were then being, shipped over, getting ready for World War I. And it continued. Everywhere the military moved, that flu hit. So we worked our way in reverse. Thankfully, we have historians who are much better at it than I am. Okay. So yeah, we were looking at the historical documents and found out exactly where it spread from. There was no other record before Kansas. But if you think, 120 people died in one city, nobody wrote something down, it doesn't work that way. Somebody's going to have a record. And we've never found anything else other than that. So once it went from the United States to Europe, it spread like wildfire because the soldiers spread like wildfire.
Yeah.
Yeah. So that's how we know.
Yeah. So the one thing I wanted to talk about is you mentioned that the technology looking for a vaccine against coronaviruses is at least 30 years old. One of the things that has kept being mentioned in those podcasts that I mentioned is that the technology, our ability to make vaccines has actually increased and sped up. So one of the reasons why, as I mentioned earlier, that a lot of people are wary of this vaccine, the SARS-CoV-2 vaccine, is that it came out really, really quickly. But one of the things that I personally believe, isn't being explained enough in the media is that just like, 20 years ago, our computing power in our computers was significantly worse than it is today. So if you go back to 2000s, a big USB was like 512 megabytes.
Yeah.
A gig was the most insanely big thing ever. We were still, and 5 years before that, we were still using floppy disks to load up our games onto the computer. So if you, it's the same thing that's happened in pretty much all technology, is that there has been leaps and bounds. So one of the reasons why that argument of it's been super quick is there's multiple reasons. One of them is that technology has improved. Another reason is that usually when it comes to a vaccine trial, at least 8 months of that is you waiting for your grant to be approved. Yes. So you've got your vaccine, and then you've just gone, can I jab this into people? And then you send out all the emails and then wait 8 to 12 months, and then coming back, it'll be like, no. So then you have to go and ask for another grant from somewhere else. Here, because we have a, international pandemic, the thing is, it's just like, we'll just make it shorter. It's like, yeah, we need to get the world back up and running. Yeah. Take all the money. You need all the money.
I wish that happened here. That'd be great.
Yeah, that would be nice. So there's that's another reason why is that there wasn't that needing to wait for 8 to 16 months or something like that just to get the go ahead.
Yeah, we honestly put almost everything on hold for this, so that when the vaccines were available, once that paperwork came in, they were hitting it right there. So It did take a little while. I do remember at least one, they said, we've got a potential candidate, and that was in March of last year, which I knew would probably take, a few months, of course, just to get into trials. It did. It took 4. It took 4 months to get into a trial. They had to prove that the vaccine would not harm anybody. They had already been using susceptible hosts long before then, but it had to be proven species specific for humans that wasn't going to cause any damage.
Yeah.
Yeah.
And Seraphim Hyde. Oh, thank you, Akita. Akita Hakubi. I can't read, but hello. Thank you for stopping by to say hello. And I apologize for absolutely destroying your name. Seraphim Hyde does say another thing I've heard is why they made a vaccine for this so quickly, but not cancer and AIDS. There's actually.
I'll talk about HIV if you'd like.
Yeah, I actually, because you've talked a little bit about how TB has affected HIV. So
so growing up in the middle of the HIV epidemic, I am probably going to get very misty. So hang in there.
Yeah, that's okay. It's understandable.
I watched, and it bothers me still. We just watched millions of people die, and they were turning their backs. They were like, oh, well, you did something wrong. No, they didn't. They didn't understand. It was misinformation. It was ignorance, sometimes willfully. Others, it was just straight up hate. Others, to me, now I'm an atheist, but I would consider the people who took care of those early on, when it was still called the gay-related disorder, those who took care of those individuals are saints to me. Like, I have a list of their names on my desk. I would like to be like that. I don't ever want the opportunity to be like that, if that makes sense. So watching all these individuals die, both men, women, gay, straight, everybody, I worried, why are we not getting a vaccine for this? I know that we've got vaccines for tetanus. I know that other things are happening. We know at this point it's a virus, all kinds of other stuff. So me internally wrestling with it and then realizing they're just not funding it. We are 30 years behind where we should be in this research because they didn't fund it. Thanks, Reagan. That's going to be your reputation. I will make sure my whole life that everyone knows the HIV epidemic is as bad as it is because of Ronald Reagan.
Yep. Yeah.
So they denied that it was happening, just like we saw with COVID.
Mm-hmm.
Except it got much worse. So the question that was asked was, why don't we have a vaccine for it? We are working on a vaccine for it. And when I say we, that is the royal we, if you will. I am not directly involved with it. It would probably make me hate my job because I would have to live through this every day.
Mm.
I carry it with me. I carry those people with me. It's something I will never let go of.
Did you lose some people to HIV? Did you?
Absolutely. Without a doubt. I know that the first person I know who died from HIV, I was in the 4th grade. So 9. I was 9. I had a classmate who was a hemophiliac, and he got it from a blood transfusion and died. Mm. People were saying, oh, it was the hemophilia. No, it wasn't. He died from HIV. Yeah. And then again, when I was in junior high, we lost 2. Like it just kept ramping up. In high school, we lost, I think, 6 altogether.
Jesus.
And then while I was in college, I started the HIV/AIDS Allies, and it was a group that just got together and was like, we tried to study it as much as we possibly could, understand each other. You know, it did not directly affect me, but it was directly affecting the people in my life, which is just as much. Like I really don't separate myself from that. Mm. So yeah, watching family members, watching friends, watching, you know, people down the street, if you will, it literally killed us. That was our whole childhood. So if you look at a Gen Xer in the United States, just mention it and watch their pupils blow up. They feel it. They see it. They saw it then. And then something happened in 92. I know that's a long time ago for some. In 1992, we came up with a treatment. The treatment moved the diagnosis, or it moved the lifespan or life expectancy from 4 years to 8. That didn't seem like much, you know, to somebody, you know, with HIV, but that was twice as long, which means our treatments were getting better. They were improving. And in the early 2000s, our treatments improved dramatically. What they were learning, all of that viral research was then going into other areas. They were learning to treat other viruses because of this one. So although I hate it with a passion, I am thankful that people were grabbing onto it. They were getting the knowledge for it, and then they were applying it in other areas. So, but in the early 2000s, we came up with treatments for it. You used 3 drugs, and you trick the virus into bringing something in. Well, the problem is with HIV, the virus becomes part of you. It becomes part of the patient. It becomes incorporated inside the DNA. It tricks the body because it's an RNA virus. It's got a protein that comes along with it. It's called reverse transcriptase. It then goes in reverse from RNA to DNA, and the cell goes, well, your DNA, you're not supposed to be out here, like inside a cell. So the cell moves that DNA that's now been tricked into the nucleus, and it incorporates into the person. It literally becomes part of you. So we can't build a vaccine just yet with the technology as we have it, because we can't attack our own cells.
Yeah.
That's the hard part. How do we kill just those? How do we make it so specific we only get one part of our white blood cells and not damage all the rest? We're just not there yet. We'll get there. No. If you want to fund something, please, from me, please fund that research.
Yeah. Yeah. Going back to that video that I did, I actually mentioned the whole lack of, basically what Reagan did in that video, because scapegoating a minority around a disease causes people to die, and that is one of them. Seraphim's Hyde said they had a scare with a cousin being exposed to it through blood transfusion. He was clear. they had another die from it, but their family sweeps it under the rug. His roommate wasn't even allowed at his funeral. That's horrendous.
I am so glad to hear that.
That is horrendous. Yeah.
That's horrific. Like, don't rip the humanity out of things.
No, don't.
I don't know what you're disagreeing with, but if you disagree with it and you're trying to hurt somebody because of it, stay out of their way.
Yeah. Yeah. Because I did an infectious disease course at university, we talked about HIV. And one of the things that they talked about that makes it so difficult to treat is that it doesn't have any proofreading capability. So people who have been living with HIV, because from what I understand, HIV, if you've been diagnosed with it, your life expectancy is roughly the same of that as somebody without HIV, which is really positive.
Yes,
that's really,
why
really
I was
positive.
Forward. When the treatments in the early 2000s came out, it moved that number from 12 to 24, and then I watched it move from 24 to 36, all the way up to now a normal life expectancy of 72.
Yeah.
So seeing it go from a death sentence in the early 80s to something horrendous through the 80s and 90s to having a small glimmer of hope in the late 90s and early 2000s to what we have now, that is brilliant.
Yeah, that's really exciting, and a lot of the like, as horrible as this disease is, and I'm not taking this away at all. In a way, it may have ended up saving more lives than the people that it's taken, because as you said, we've learned a lot more about viruses because of this particular virus.
Yeah.
We've also learned a lot more about the way our immune system works. We've learned a lot, and I think they're using some of the information that they've learned from HIV is a way to use towards cancer research.
Yes.
So. And also, I was talking to somebody who was, basically the huge chunk of their lives was, his life was, because he was a gay activist, but he was also an AIDS educator. And he mentioned that one of the things about AIDS is that it normalized people being gay, because during the AIDS epidemic, people were coming down with AIDS and they had to come out and say, I'm gay. And then all of a sudden, gay people went from being icky, yucky people to my son, my brother, my actor, and this actor, like Freddie Mercury. And all of a sudden, he's like, people are now going, okay, these are genuine people that I genuinely love. And then all of a sudden, being gay stopped being so bad.
Yeah.
So,
it changed their cultures in more than one way.
Yeah. So it's a horrible disease, but there's a lot of positive things that have come out of it. So, there's always a silver lining. Going off something a little less depressing, Beach Price asks, I'm sure Dr. Hobbs has looked at a lot of microbes. Which one does he think looks the prettiest?
Anything is amphitrechous, which means it has multiple flagella. The flagella go in like every direction. And when the microbes are moving, they kind of bind into themselves and then they go in the opposite direction. I love it because you get to watch them go. So any of them like that, I love it.
I can't remember the name of the agar that we used, but it was a selective agar that when you put E. coli on it, it grew into this black metallic sheen. Do you remember the name?
Yeah, the green sheen. Yes, it's EMB, eosine methylene blue, EMB plates.
Oh, they're gorgeous.
They're beautiful, yes.
Yeah.
And only coliform bacteria will do that because they have to do it rapidly, and it makes it turn kind of a metallic green sheen. And you can't describe it. Like I said it just now, and you think, look it up. Like really go online and type in EMB metallic green. It really looks like that. Like it's not CG. It really does look like that. It's beautiful.
It is. It's almost, the sheen, the metallic sheen you get on bugs.
Yes.
Almost. Yeah, that's absolutely gorgeous.
The green shiny bugs. I didn't even think about that. Now I will do so.
Yeah, because when I saw it was so pretty. Also, Esmarsensens, I think that's how you pronounce it. It's a microbe that it causes that pink goo on you. But when it's isolated and grown in agar, it's bright red and it is beautiful. There are some gorgeous microbes out there.
There are some microbiologists who are also artists, and they paint with microbes. They know exactly which microbe to put where so that it shows up as a picture. That is awesome. Everyone, you need to look up agar art. It is stunning. Some people are really amazing at what they do. Yes, I concur.
Yeah, and there's like genuine artwork in that. Oh, it's gorgeous. Do you want to talk a little bit about bioremediation? Because you also talk a little bit about that, because I get a little bit excited about bioremediation as well.
So what bioremediation is? The use of a microbe to clean up any type of pollution. Like you just pick up pollution, we're going to try to find a solution for it using microbes. Microbes that can be selected in or selected out depending on the environment. A good example would be something like an oil spill. You don't want all of that crude just sitting there. There's no real way of trapping it all. There's waves and other types of elements that would cause it to get out. So what if we developed a particular bacteria that you could then spray as an aerosol that wouldn't harm the environment, but would then damage that crude so that you turn it into something like either methane or CO2, whatever, some other product, something either non-toxic or something that becomes gas, like a gaseous agent. So we can remove that directly from the water so it doesn't get on our beaches and so on. Bioremediation is not just for liquids and things like that. We can also use them to get rid of things like radiation. There are microbes that we can spray on radiation or put near radioactive sources, and they'll either protect the rest of the environment from the radiation because they can handle it, or they can get rid of it completely. Thinking about Chernobyl from '86, knowing that all of that went into the atmosphere, toxified Ukraine. So you took the breadbasket of Europe and you toxified it so that you can't really farm it. It's still radioactive, and it will be for the next 3,500 years. So if we took a microbe, spread that into the soil here or there, it will naturally die off, but it's going to remove the radiation from it first. We then take that toxified farmland and turn it into something that we can use.
Yeah.
Microbes are amazing.
They are. I keep saying to people that I think microbes are going to save the world. I genuinely believe that. Because the microbe that you were talking about has like the mechanisms to how they protect their DNA from being damaged is just weird. Like microbes are weird. They're really weird.
Go ahead.
No, you go because I only.
They also replicate really quickly. So if one's damaged, there's 7 billion others just sitting there waiting.
Yeah. One of my favorite microbes, other than G. kombucha, because G. kombucha can, as I said, can be used as bioremediation as well, but is something called Ideonella sacchaensis, and it's a microbe that produced a enzyme called PETase. Which destroys PET. So if you don't know what PET is, it's basically anything that's plastic. So this is PET, this is PET. So, and as we are all very well aware, PET, plastic pollution, is terrible, absolutely horrendous. But Ideonella sacchaensis has been able to produce a enzyme which is actually a fairly new enzyme because scientists accidentally improved the speed of being able to break it down. So if you don't know what an enzyme is, it's a protein. So all enzymes are proteins, but not all proteins are enzymes. And an enzyme basically is a protein that produces that lowers the energy required to start a reaction. So, for example, you've got lactase, which is a protein that breaks down lactose. If you didn't have lactase, your body wouldn't be able to break it down, which means that you are lactose intolerant. And anyway, so what happens is this enzyme just breaks apart the PET, meaning that we can possibly use Idionellosacchensis, a genetically modified version that is much better at with that more effective enzyme being able to just help get rid of microplastics and things like that.
Yes.
So that's exciting, and that was found in a plastic plastic processing facility in Japan. So yeah, that one's really cool.
Yeah, microbes are amazing. Every time I learn about another one, I am just amazed. I don't know why I'm amazed. They've been here since 3.5 billion years ago. We think we're special. We've been around for what, 4 million years?
Something like that.
Yeah, that's nothing.
One of the things that always blows me away is it's like, if there's a problem, there's a microbe for it, and if there's a problem, it's usually a microbe causing it.
That's it. I'll focus on that. Yes. So the BCG, which is the vaccine for tuberculosis that doesn't work very well, that doesn't mean don't get it. If children are born in high TB populations, they should get it. We just don't give it to adults. It doesn't really work. But what we have found is that it works really well for treating bladder cancer.
Oh, wow.
So silver lining for tuberculosis. I only want to take care of the human pathogen, not the stuff that kills off the cancer.
Oh, wow. Well, so TB kills the cancer, or the vaccine causes it?
What? The TB itself, not pathogenic TB, one that comes from a cow. It's the, I don't want to say attenuated, even though it relatively is. It's a weakened version of TB, will kill off bladder cancer.
What?
Yeah.
Jeez.
It knows how to infect the cells, and it's really good at getting into those tissues, damaging those cells. The cancer cells die off or slough off. There's nothing else to eat, and you just urinate and it comes out.
So how do they like inject the microbes into the bladder?
Yep.
That's crazy.
So more research, of course, is needed, but yeah, it's showing very good efficacy. It's working really well. So you can look that up. Go ahead.
I need to process that. That's oh, wow.
Yeah. So for every problem, there is a bacterial solution.
Usually,
Dental caries, we're doing that with animals now. It has been approved for animals. The lactobacillus that causes the lactic acid on our teeth that causes tooth decay. In dogs, we can give them a secondary type of lactobacillus that will displace the one that produces lactic acid. So it can't damage the teeth, but it keeps the bacteria present. So something else doesn't get in and overpopulate. So you put in a bacteria, you swirl around their mouth. It displaces the bad. They keep the good. They keep all the benefits from it. Microbial antagonism. So they don't get other types of infections, but they don't get the tooth decay.
That's cool. That's really cool. So is that only in dogs, or is that just in trials in dogs and hopefully getting into?
Well, right now, it has been approved to use in dogs, but we haven't been approved to use it anywhere else yet.
Okay. That's cool. That's cool.
Yeah.
There's a lot of I was listening to a podcast. If you're interested in microbiology as well, there's a podcast called TWIM, or This Week in Microbiology, and it's run by the American Society of Microbiology, and it's very, very interesting. And they were talking about how microbes produce these things called vipers or vipers or something. I can't remember what it stands for. And it's basically the microbe has its own immune system. So, you know, if everyone's heard of CRISPR, like CRISPR-Cas9, basically what that is, it's like a molecular scissors that chop up things, chop up DNA. And vipers have a very, very similar in the way that it can kind of just like it's an immune system. I'll have to listen to this episode again, but if you want to look into it, it's VIPIRS. Microbes have their own immune systems, and we are able to use those the immune system as a way to produce antiviral medication. So that was one branch of us trying and figure out the insanity of the COVID-19 as a treatment, not as a preventative thing. Did you want to actually talk a little bit about the Cas9 protein, because do you know much about it?
As much as I have read about it, I have to still say no. There is a mountain of information. I would like to know more. There's a specialist somewhere else that knows a million times more than I do. So I know just enough to get myself in trouble. And somebody who knows a little bit more will say, So I'm going to say no. But if you have it, I will be happy to discuss the outcomes with you. Yes.
Yeah, I don't know too much about it because it's incredibly complicated. But basically, the Cas9 protein, Cas9, it's basically a molecular scissors, and they come in and just snip up the DNA or RNA, and then they can they've been able to figure out how to do it at particular points, and then they can clone and do all of these crazy things with it. So it's one of those tools that once it became available, it was like our technology just went just went insane.
So late in the 1970s, when we started to develop the humulin, that's the human insulin, we used bacteria to do that. And the technology that led up to this that we're talking about came from that research. So what we have are restriction enzymes. Now cut the DNA in a certain area. We then apply another gene to that and then allow it to reattach. The microbes will then go through their normal physiology and they'll make the product that we're looking for. So human insulin, the injectable human insulin now comes from a microbe. All we have to do is feed it and keep it safe, and it will work forever. Which bothers me because you have to pay $300 for it here in the United States.
Yeah, that's not okay. Yeah.
For the same type of product that we use to make cheese and soy sauce.
Yeah, it's not okay. It's the joys of capitalism in a thing that shouldn't be capitalized.
We're working on it.
Giant Butthole does say, twice in 24 hours I've seen people claiming the COVID vaccine uses CRISPR or something like that to insert genes into our DNA. It's the latest conspiracy theory. Would you be able to address this insanity?
Yes. No, that doesn't happen. The vaccine has already been made. When it's injected, how are they going to do that? You have to raise it and lower it. You have to raise it and lower it, talking about the temperature, and our bodies don't do that. No. So I'm not trying to be a smartass, but basic physiology would tell them that can't happen.
Maybe they might be coming off the whole idea that, as you mentioned earlier, with HIV, it actually enters into the cell, the cell's DNA. No, it's not how it works. Would you be able to?
I'll talk about that.
Yeah, would you be able to explain?
RNA viruses, when they get into a cell, they go directly to the ribosomes generally. Okay, there are multiple types. So if you're a strict microbiologist, hang in there. So the RNA gets in, they go to the ribosome starts making these products. Okay, so all these proteins and new parts of the virus will then be assembled inside the cell. It's either going to slowly release it or it's all going to burst open and just infect the next cell.
Would you be able to explain what a ribosome is?
A ribosome,
Very
yeah,
quickly.
sure. It's a very small sequence of RNA, which is a copy of the DNA that knows how to read RNA so that it can make a product. So basically, it's a reading enzyme so that we can take the code from where we need it and make the product that we're looking for. Does that make sense to everybody?
Yeah.
Okay. If not, I'm doing it another way. But ribosomes are found on the inside of cells. Sometimes they're attached, sometimes they're free. So a virus will get in and start making those proteins, those products. What we try to do with a vaccine, oftentimes, is make a product of what will be associated with a virus, such as the spike. We train that particular cell to say, all right, this protein is bad. Present it to all of your antigen presenting cells. So all of your cells, or then your immune cells, are going to learn this is bad. I'm showing it to you. This is bad. You see it? They're like, yeah, I got it. I now have the code. Kill it. If you see it, kill it all, because this is bad. And that's what the mRNA vaccines are trying to do. We don't give any of the actual virus. We give a code for a part of the virus. Well, that part is the important part that sticks up. It's like, hey, I'm virus. Anything that's got that, we then attack and kill. That is a very overly simplistic version, but at no point in that overly simplistic version did we mention that RNA then goes into the DNA or anything like that. It doesn't work its way in reverse. There's no reverse transcriptase. Even if there was a reverse transcriptase, it wouldn't make viral proteins. That's one of the benefits to it. That's how we know that it doesn't work that way. But once we have that spike immune system built up, they'll talk to each other. The cells really do communicate with each other. B cells will talk to T cells will talk back, and so on, so that they know exactly what invader to fight off. Okay, does that answer the question effectively to everybody?
I think so. Giant Buttonhole does say perfect, thanks.
Sure.
Beach Price does say, will our bodies be making fragments of ERVs to pass on to future generations for being infected with this virus?
No, unfortunately. When we become infected with something, we have something that's called adaptive immunity as compared to innate immunity. Innate immunity, natural killer cells, the antibacterial proteins we have in our skin and so on. Our skin itself is antimicrobial. It keeps things from getting in. That can pass to somebody else. Like if you've got good skin, you're probably going to pass that to your kids. But we're probably not going to be able to pass that from one person to the next generationally, because that information doesn't become incorporated in our DNA. And since it can't pass to the next generation. That's why we have adaptive immunity, and that's why we have vaccines in childhood. What we are working on now is a childhood coronavirus vaccine. It's most likely going to be the same that we have now, but it takes a lot of paperwork, mountains of paperwork, to be able to get that through so that you can work on children who cannot volunteer, and you can't volunteer somebody else. So it's questionable how that works. So there's a little bit of a gray area there. I will see, and I'll see if I can post it on how those vaccines actually get approved. I don't know all of the legal ramifications or all the legal requirements, because I've never done a childhood vaccine.
Yeah.
So I don't want to speak out of turn.
Fair enough. All right. Well, we're coming up to about the hour and a half mark. If anybody wants to ask any questions towards Dr. Stephen Hobbs, please let me know in the chat. But a couple of questions I do have for you. So can you tell us a little bit about your education, your past in regards to...
No, I don't want to talk about myself. So I started as a biologist and chemist and just fell in love with it. I could not get enough. When I went through chemistry, I realized that most of the stuff that I was using was physics. Like really, if you want to study how a particular chemical looks in space, like the 3D structure, you put it through an NMR. The advisor that I had, and I love him to death, I always have, and I always will. He will always have a soft spot for, in my heart. He let me break the machine kind of on purpose. Not really, but he was like, yeah, I need you to take it apart, but he knew it was going to break. I need you to take it apart, broke it, and fixed it. I'm more of an engineer. I am not an engineer. Everybody knows this, but I have a mindset. Engineering can fix almost everything. Like engineering, math, I will always like the tip top cream of the crop sciences for me involve engineering and mathematics. Like I'm biased that way. So he let me take all of this stuff apart, and then he said, "Okay, I need you to build one." I have no idea what I'm doing. I have no idea what I'm doing. He said, "Well, here's the schematics. Take off." All right. And he gave me a budget. And I was able to fix the NMR from being broken. It didn't run. The machine itself did not work. He knew it didn't work, and I got it to work. So we hooked it up to a computer, a couple of rooms away, because you can't have a computer in the room where the machine is. And it worked. He was kind of blown away. I certainly was. I'm like, I can't believe this worked, but I followed all the schematics to go along with it. So he was like, you're pretty good at this. What else do you think you can build? I was really interested in Rosalind Franklin because I was learning about Rosalind Franklin in biology. So I wanted to know how X-ray diffraction worked. So he said, well, just build one. What? Are you crazy? No, I'm going to just build it. It can't be that difficult. Look what you've already done. He knew what he was doing, but I didn't. So I built the University of Texas Permian Basin's first X-ray machine. It's still there, and they still use it.
Wow.
Well, yeah. Biology and chemistry, and to a certain extent physics, became part of me, and I have never let that go since. Because I know that if I want to study biology, I have to know chemistry. And if I want to know chemistry, I got to know physics. When you're studying physics, you end up studying basically chemical history. Like, how did all this work? How did they detect this? You hear about scientists going up in the air and they've got their little fog box so they can see the particles shooting through. How did they know about the fog? That's all chemistry. So if you want to study one, you have to study the other. And then I just kept going. Like I couldn't get enough. I had to go. I knew I wanted to be a doctor. I knew I wanted to take care of patients. I knew that I wanted to keep, I wanted all of it. I'm kind of greedy that way. I want all of the knowledge possible so that when I die, I can say, hey, I knew about that, or just before I die. I fell in love with it. Treating patients was fine. I had practice for a while. I missed teaching because I started teaching as I was going through school. I started teaching gross anatomy. My colleagues, my other students, or the students that were going to school with me, were asking me, what is that? What is this? Like, I ended up teaching them. But once I went into practice, I missed the teaching aspect. So I called one day and it says, it was from a previous patient. He didn't tell me who he was or what he did. He just said that he worked for the college. It was a chancellor. And he said, I have a job for you if you want it. You really helped me, and I think you can help, the program. And I've been teaching ever since.
Nice.
So that's my background. I mean, it's kind of boring when you hear it, but I mean, it took me forever to get through there, and I haven't regretted a single step.
Oh,
I would do everything else exactly the same.
Nice. I've got a couple of questions. How are the kids from Beach Price? How are the kids?
Oh, my kids are great. They are all at home. They get to study at home through online learning. And I mean, they have me as their dad. So as good as they can be with this. So, but they put up with me pretty well.
Lassus Vulgaris asks, how likely is it for another pandemic to appear in the near future?
With these variants, highly.
Yeah.
Like I don't want to be doom and gloom. If the variants respond to the vaccine, but we're not getting the vaccine, we have a problem. If people get the vaccine and the variants are still spreading around, it can still come back, just like the Great Flu of 1918. It'll make its way around. It went around the world 4 times.
And we're still... We're still...
Yeah.
With the 1918 influenza virus, we're still have it in our population. It's just now like the yearly flu. yeah, it's highly likely we're going to have it a couple of different ones.
Another one that we worry about is lysoviruses, or the family of rabies, if you will, and then the Marburg viruses. So kind of like Ebola. We worry
And
about
also-
those a lot.
and also smallpox, because of climate change is coming back. There's a possibility that smallpox will also come back because yay!
But now we know how to make the vaccine at home, just in case.
Yeah. Actually, there is still companies producing vaccines for the
Armed forces definitely do them.
what's the name of the virus family? I keep forgetting it for the smallpox. Is it?
Oh, crap. Orthomyxoviruses. Is that right?
No, I don't think it's that one.
Doesn't seem right. That's a flu.
No, I can't remember either.
You think I'd be better at this? I'll think about it.
Mama Atheist goes, how do you see math? Or maths?
How do I see math? Math? Like the study of numbers?
Yes.
Yeah, we talked about that not too long ago.
Yeah. She does say his answer is beautiful, so I apparently
the
have
question
to.
was
I
thrown
watched
at
it.
me, and I appreciate. I took a little bit of time to think about it. It said, was math invented or discovered? And I don't agree with either of those. It was recognized. Like everything is a symbol. Math is a symbol for, how many? If you've got 4 fingers, you have 4, so we write down the symbol. We recognize that in our head as a symbol, and then we can use those symbols to make pretty much everything. Math is everywhere. It's art to me. It's poetry. I argued with somebody on Twitter, apparently a very well-known artist, and I did not know that. Sorry. That math itself is artwork. When you see a formula, it's beautiful. Like when the formula works, regardless of whether it's something very complicated or very simple, like force, mass, acceleration, so on. What that entails and what that shows, all of the ingenuity behind it to recognize what that is, and then to be able to disseminate it to others so they see it exactly the same way you do. I can't think of anything more poetic and artistic than that. So we recognize the beauty behind it. Math is recognized rather than invented or discovered.
Yeah, it is beautiful. I am going to wrap this up because, as I said, it's been about an hour and a half. And thank you very much. Thank you for nerding out with me around microbes. I mean, we could honestly talk for friggin' hours. Yeah, there's microbes are just amazing. They are genuinely amazing little critters. They are very underappreciated, and and I do really appreciate you coming on.
You're very welcome.
I just want to say thank you for everyone who's come in and continued watching. I really appreciate that. I'm very grateful for that. And you said you've got a Twitter account.
I do.
And you said you've got a very, 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 very small YouTube channel.
Yeah. I mean, it's there if you want it. I've uploaded like 5 videos.
Yeah.
But I typically tweet.
Yeah. All right. Well, is there anything that you would like to say in conclusion, or?
Yeah, well, thanks for having me. I've appreciated the opportunity. I have enjoyed all of the conversation. Thanks for having me.
And I just want to say thank you, every single person, for coming. And don't forget, don't give yourself the raw prawn.
And it is orthopox virus, by the way.
Orthopox virus. Okay, thank you.
You're welcome. See you later.